Hello students, so welcome back to the channel. In this video we are going to study about the electron transport system and oxidative phosphorylation. So if you know key in all the respiratory process, whether it is your glycolysis or link reaction or Krebs cycle, the energy is stored in NADH plus H plus and FADH2. Is it clear? Now we will oxidize it through the process that is known as electron transport system. Is it clear? They are oxidized through the process of electron transport system, and finally the electron is given to the oxygen. The oxygen is an ultimate acceptor of electron. It will result in the formation of water molecule. Clear? Now the pathway in which electron will be passing from one carrier to another, that is known as system, that is known as electron transport system. And this system is present on inner mitochondria membrane. So this is again one question area ki where does ETS occur? Okay, ETS means electron transport system. Where does it occur? It occurs in the inner membrane of mitochondria. Is it clear? Now electrons from NADH plus H plus, which are produced in the mitochondria matrix through citric acid cycle. So we have already studied in citric acid cycle, which is also known as Krebs cycle. NADH plus H plus are produced in mitochondria matrix. Is it clear? So they are oxidized by a first complex that is known as NADH dehydrogenase. Is it clear? So NADH is getting oxidized with the help of an enzyme NADH dehydrogenase. Up till here, is it clear? Now let me explain you with the presentation on the board. Now see here, try to understand if any substrate is changing into the product, so it is giving its hydrogen to NAD, which is forming NADH2. Is it clear? They should touch NADH. 2 formation is going on. So you know very well that it is NADH plus H plus. So let me write it full for you. And this is NAD plus. So when it is getting oxidized back, it is giving its hydrogen to FMN. And this FMN, after taking hydrogen from here, it will change to FMNH2. Is it clear, beta? Now it is getting itself oxidized. So this is a set of one enzyme. This is your one enzyme, and this enzyme is known as NADH dehydrogenase. So this is your complex one, which is known as NADH dehydrogenase. So it will give the electron to next member, that is your ubiquinone. It will give it to ubiquinone. Okay, system is giving only electron, and it will remove and accumulate the proton in the intermembrane space. And only electron is given to ubiquinone. But ubiquinone is not an electron carrier, it is a hydrogen carrier. So it will remove the proton from the matrix. Because system is giving 2 electrons, it will remove 2 protons, and it will become reduced form of ubiquinone, that is known as ubiquinol. Ubiquinol. Is it clear? So up till here concept is clear ki complex 1 is oxidizing NADH and giving it to ubiquinone. So you can see here, see one thing, your NADH is oxidized by NADH dehydrogenase complex 1. So you can see the board carefully. This is your complex 1, NADH dehydrogenase. See, NADH is changing into NAD plus, and it is giving to this yellow color. You can see FMN is present in it, enzyme. And if you see, it is accumulating proton in where? It is accumulating proton in intermembrane space. Please focus, beta. It is accumulating in the intermembrane space and passing the electron to the ubiquinone. Okay, ubiquinone. And ubiquinone is then becoming into ubiquinol. So up till here concept is clear. You can check the arrow pointing is going this way. And now see carefully. Now it will get oxidized. So when it is getting oxidized, it is giving its electron to next, that is your cytochrome BC1. Okay, here iron will be present, Fe3 plus, 2 electron will be given. So here your cytochrome B, this is Fe2 plus, it is BC1. Because cytochromes are rich in the iron, so when the system is giving electron to them, so they are also removing the proton to the membrane. So it is removing the proton to the membrane. So this is your complex 3. This is your complex 3. Complex 3 name is your cytochrome BC1 complex. Now, up till here concept is clear. Now focus very carefully. If you remember in the Krebs cycle, FADH2 is formed when the succinic acid dehydrogenase enzyme. So FADH2 when changes into FAD back, it is giving its entire hydrogen to ubiquinone. FADH2 is giving the entire hydrogen to ubiquinone. So here your complex 2 is present. That complex 2 name is succinate dehydrogenase. Succinate dehydrogenase. So from the diagram, up till here concept is clear. Ki complex 1 is giving to ubiquinone, even complex 2 is also giving to ubiquinone. And ubiquinone is then giving to complex 3. Now see the diagram. Okay, so NADH dehydrogenase is getting oxidized and electron are then transferred to ubiquinone, which is located within the membrane. Please learn this location question also. Ubiquinone also receive reduced equivalent by FADH2, that is complex 2. The ubiquinone also get from complex 2, which is generated during oxidation of the succinate in citric acid cycle. And reduced ubiquinone is known as ubiquinol. This is your question area. Reduced ubiquinone is known as ubiquinol. Is it clear, beta? This is important, please. Is then now it is getting oxidized with the transfer of electron to C by BC1 complex, that is complex 3. The wait here, this I have to explain, but you have understood here. Your complex 3 is cytochrome BC1 is your complex 3, and your NADH dehydrogenase is complex 1, and your succinate dehydrogenase is complex 2. Check the diagram carefully. See here. This is your complex 2. Check complex 2 succinate dehydrogenase. So when succinic acid is changing to fumaric acid, it is giving to FAD, you can see carefully, and it is check the arrow's orientation. It is also given to ubiquinone, which is getting reduced, and they both are giving to complex 3. From this side, ubiquinone is giving to complex 3. Even from this side also, ubiquinone is giving to complex 3. That is cytochrome BC1. BC1. Is it clear? You can analyze the arrows carefully. Electron is going here and they are removing the proton. The proton is removed from complex 1, proton is removed from complex 3. And electrons are transferred from BC1. Now see the diagram carefully. Now see the next step. Next is your cytochrome C. This is your cytochrome A. In the entire system, only electron movement is going on, we have to remember. Okay, and last is your cytochrome A3. So system has made the electron reach to here, and in the last here, if you focus, please focus carefully. Here you will find hydrogen is removed from the matrix and half oxygen is coming. Half oxygen is coming. So you know very well, proton plus electron will combine to form hydrogen, and half oxygen means they will combine to form water molecule. So you know very well, when two protons plus two electrons plus half oxygen, then I think so total formation will be H2, one hydrogen molecule, and half oxygen means one atom. So water molecule is formed. So this is your complex 4. Complex 4 is your cytochrome AA3. This is your complex 4, that is cytochrome AA3. Is it clear? Now see carefully. So they are telling it, check carefully, the reduced ubiquinol is then oxidized by transferring the electron to C by BC1 complex. So this reduced is giving BC1, carefully check the diagram carefully. Cytochrome BC1 is getting oxidized by giving electron to cytochrome C. Check carefully, it is giving to cytochrome C. Is it clear? And from cytochrome C, it is now coming to complex 4. So cytochrome C is a small protein attached to the outer surface of inner membrane. You know very well this process is occurring on the inner membrane of mitochondria. You can see here. This process is occurring on the inner membrane of mitochondria. And you know very well cell membrane is phospholipid bilayer. So this is on the outer surface of inner membrane. Please learn carefully. It is present on the outer surface of inner membrane. It is a mobile carrier, electron carrier for transferring of electron between complex 3 and 4. So what is your complex 4? Complex 4 is cytochrome C oxidase. You know very well why we say complex 4 is cytochrome C oxidase, because it is going to remove electron from cytochrome C. And it is AA3 with 2 copper center. Focus the diagram. See here. This is your cytochrome, okay, A3, and it is having 2 copper center. So up till here, I think so concept is clear. If you check the diagram, we have discussed this is our complex 1, NADH dehydrogenase. This is our complex 2, succinate dehydrogenase. Then our complex 3, cytochrome BC1. Then is our complex 4, that is cytochrome AA3. This is the one ubiquinone is the one which is taking from the 1, giving it to 3, taking from 2, giving it to 3. Is it clear? And ubiquinol, it is they are saying in the NCERT, ubiquinol is getting oxidized by giving its electron to C by BC1. So sequence is clear na? BC1, then it is going to give it to C. So this is your mobile carrier. Cytochrome C is mobile carrier between 3 and 4. So now see here. So when the electron, they are passing from one carrier to another, that is from 1 to 4, in between, they are coupled with the ATP synthase, that is complex 5, which is responsible for the formation of ATP from ADP and inorganic phosphate. So you can see here, this is your complex 5. Okay, this is your F0 particle, this is your F1 particle. I hope you are able to understand my diagram. Where am I? I'm here on this diagram. Okay, beta. So this is your complex 5, which is in the head. It is having an enzyme, that is your ATP synthase. So this is your complex 5th. So complex 5th is what? ATP synthase. This is your complex 5th. Now, number of ATP, how many ATP molecule will be synthesized? It all depend upon who is the electron donor. One molecule of NADH, please focus, one molecule of NADH give rise to 3 ATP. While one molecule of FADH give rise to 2 ATP. So who is the electron donor? It all depend upon that. NADH will give rise to 3 ATP and one molecule of FADH give rise to 2 ATP. Now see, although the aerobic process of respiration takes place only in the presence of oxygen, but where we study the role of oxygen is only in the terminal stage of process. You can see in the diagram, check the diagram carefully. Check, oxygen is used only in the last step of ETS. You can see here oxygen is used here only in the last step of ETS. Clear, beta? So, yet the presence of oxygen is vital. Why it is necessary? Because it derives the whole process. It means it is making the process to work continuously. The no halting is coming. How it is making the process to work continuously? By removing the hydrogen from the system. So we can say oxygen act as a final hydrogen acceptor. Oxygen act as a final hydrogen acceptor. Now, one more thing I just want to highlight here. Just give me 1 minute. Now, beta, see here. Check carefully. Oxygen is the ultimate acceptor of electron and it is reduced to water. This is the line given in our summary area. Okay. So oxygen is an ultimate acceptor of electron and it is a final hydrogen acceptor so that it can make water. And we say oxygen is reduced to water because you know very well when anything take hydrogen, we call it as it is reduced. Is it clear, beta? So now see carefully. In the photosynthesis, it is in the photosynthesis, light energy is used for the production of proton gradient. So in photosynthesis, that is why we are saying photophosphorylation, light energy is used to for the creating the proton gradient. But in case of respiration, we have to focus from here. In respiration, it is the energy of oxidation and reduction because we are oxidizing one thing and we are reducing another thing. So oxidation and reduction energy, okay, it is the one which is actually helping in creating the proton gradient. So we are creating the proton gradient. That is why this process in which we are creating the proton gradient and then we are breaking it. Okay, this process is first we create the proton gradient and then we allow this proton to pass through this F0 F1 particle. You can check the diagram once again. See here. See, the proton gradient is established in the intermembrane space. See here, 4 proton here, 4 proton here, and 2 proton from the complex 4. And finally, this gradient, if you see here, this is a plus sign. Check carefully. Plus sign. And here it is minus sign. Why it is plus sign? Because too much proton is accumulated here. And here it is minus. So electrochemical gradient is created. And this gradient is broken. See carefully. These protons are allowed to pass through F0, F1 particle. So when they're allowed to pass through F0, F1 particle, then ATP formation is done. Clear, beta? So that is why this process is known as oxidative phosphorylation because we are using oxidation reduction energy to create proton gradient. So now see one thing, chemiosmotic hypothesis. Energy released during the electron transport system is used in synthesizing ATP with the help of ATP synthesis, that is complex 5. See this diagram. If you see, this is F0 and this is F1. So F0, see F0 is an integral membrane protein. Please, it is an integral, meat is totally embedded inside. Integral membrane protein, which forms a channel through which proton can pass. It is working as a channel through which proton can cross the inner membrane. Okay, now F1, check carefully. F1 is headpiece, it is a peripheral membrane protein. So now focus the question, which is integral membrane protein and which is peripheral membrane protein. So F0 is your integral membrane protein and F1 is your peripheral membrane protein. And this one, which is peripheral membrane protein, it contains the site for synthesis of ATP because it is having the complex fifth, that is ATP synthase. Is it clear, beta? So the passage of proton through the channel is coupled with the catalytic site, and for each ATP formation, when you are making one ATP, two protons have to pass. Is it clear? And they are passing down the electrochemical proton gradient, which I have already shown in the diagram, how electrochemical gradient is there. So come back to this slide again. If you see, four protons are accumulated here, four protons are accumulated here. Even this is also accumulating two proton. Okay, two protons it is accumulating, it is removing two protons. So this is the way how proton gradient is created, and it is then broken down by allowing through F0 F1 particle. Is it clear? So I hope this practice, which is right now this presentation on the board, this is helping you to learn the name of the complexes. And I made you read the theory of the NCERT line to line. I hope this topic is now easy for you. Okay, thanks for watching.